Okay, welcome to chapter 11, and I will get my pen going, and I will turn on my timer. Basically, in this chapter, we're going to look at how genes are controlled. Big thing in biology is basically looking at when genes are turned on and turned off. And then during the, just kind of as a size, during the 1900s, doctors noticed that smoking increased and that lung cancer had increased in this picture of metastases in the lung. It wasn't until 1996 that research studying lung cancer found that, again, a component of tobacco smoke binds to DNA within a gene called P53, which codes for a protein that normally helps suppress the formation of tumors. So if you press the tumor suppressor, tumors occur. This is the original smoking gun for the cause of cancer. So where we want to go is where we want to look at is how and why genes are regulated. And hopefully you do know that every somatic cell, that is, every cell in an organism contains identical genetic instructions. And so they all share the same genome. So then what makes them different? And this is a process of cellular differentiation where cells become specialized in their structure and also in their function. So an eye cell is an eye cell, and muscle cells are muscle cells. Certain genes are turned on and off in this process that we call genetic regulation. In gene expression, a gene is turned on and transcribed into RNA and then goes into proteins. And so we remember something that we call the central dogma, where genes get turned into proteins and your genotype gets turned into a phenotype. Information flows, for the most part, from DNA to RNA to proteins. There are some exceptions, but that's the big one. And so, again, what happens is that the big differences that we see among cells in an organism results in what we call selective expression of genes, and that different cells will produce different genes. And so here we see in a pancreas cell, we have genes for the glycolysis enzyme, no antibody genes, no hemoglobin genes. In a white blood cell, we will see some of these genes in a nerve cell. And so it's this different pattern of genes that will give us these different cells. Gene regulation in eukaryotic cells. Ideally, I'd like you to read up about the operon system in prokaryotic cells. But in eukaryotic cells, we see much more complex genetic regulation. And there's a number of different reasons why. And basically, we have the genes that are in the chromosomes, and these chromosomes are folded up in during interphase sometimes, and they can affect genetic expression. So that's why we hear of this here, unpacking of DNA. And so once, in order to express a gene, you need to expose that gene, and in order to turn it on, we then need to transcribe that gene. That gene then needs some processing. So when we make the messenger RNA, we put a 5' cap on it, a 3' tail. This then needs to move into the cytoplasm, where it will actually get translated. This messenger RNA can also get broken down and has a certain half-life of it. Once we make the peptide, it doesn't always stop there, but the protein itself needs to fold up into an active format, and the net protein can be broken down either quickly or not so quickly. And so there's lots and lots of genetic regulation that we will see there. And so, again, let's look at packing. The example that we have of packing is basically occurs in female mammals, where we have basically two X chromosomes. One of those X chromosomes is inactivated relatively early, and so all of the descendants will have the same X chromosome turned off. If a female cat is heterozygous for a gene on the X chromosome, about half her cells will express one allele and one half will express another allele. And so what we see in cats is that in this chromosome we see that we have a chromosome for orange fur and we have a chromosome for black fur. And depending on which of these X chromosomes are turned on and off will give us a different coat color in the cat. And again, we can see this in the cats. RNA processing and breakdown in eukaryotic cells. We see the localized transcription in the nucleus and the process of RNA in the nucleus. The RNA processing is the addition of a cap and a tail to mRNA and removal of what we call the introns. We'll talk about this in the splicing together of the remaining exons. And so what we call alternative splicing. So let's say we have a gene, and this gene we may have a number of, we have what are the exons, and these are the introns right here. And what happens is that we can express and put different exons together in order to give a different functionality of that gene. Again, depending on what RNA we splice, we can get this protein over here, or we can get this protein, this type of protein over here. It's not a big, huge change, but it is a change nonetheless. Okay. Again, messenger RNAs can last from hours to weeks to months, and eventually they're broken down and recycled. And then I'm not going to get into that. Cell signaling hormones can have an effect on which genes are turned on in a cell. And so here we see a signaling cell produces a hormone. That hormone then can interact with a membrane receptor in the plasma membrane. That will go through signal transduction pathway, and it will have a response in the nucleus. And so again, cells can actually modify and regulate what's going on in other cells. How we look at this, one of the ways that we look at this is what we call a microarray. And so we can look at the messenger RNA that's produced by certain cells by purifying that messenger RNA, by labeling it, and then binding it with what's called a DNA microarray. And this is a small sheet of glass that has been exposed to, in this case here, over 6,400 different genes. And the amount of messenger RNA in here can tell us whether this thing is kind of turned on or off, and that's important. We'll also look at cloning of animals. And again, we've done the cloning in our laboratory of rice. In this one here, we're talking about the cloning of the carrot. If we take root cells from the carrot, we could differentiate those root cells, and we can make a new organism from that root. And again, this will give us many possibilities. Plant cloning demonstrates the cell differentiation in plants does not cause irreversible changes in DNA, and it's now used extensively in agriculture to make the type of plants that we want. Now, in mammals and in animals, it's a little bit different. And again, we've seen the regrowth of the legs of the salamander, of the tails of the salamander, and so we thought that maybe cloning might be possible in animals. And one of the things that we do in order to clone in animals is this process that we call nuclear transplantation. And again, this was started in 1997. What we do here is we remove the egg from an egg cell. We add a donor cell nucleus, and then this nucleus from the donor cell, then if we allow this egg to go through the differentiation process, will produce a new organism. And again, it has to be born, it has to be put in the uterus just like the mother. And here's an example of the first clone that we saw, Dolly. Other mammals have since been produced using this technique. Mainly they're farm animals. We're very interested in farm animals because, again, we're interested in producing animals that are genetic to one another. Again, people who really love their pets may clone their pets. Again, farm animals that produce organs that look very similar to human being organs that we're looking at. So, again, endangered animals that we do not have a lot of have been cloned as well. Therapeutic cloning is not necessarily what. So, again, what we're doing in stem cells is we're looking at cloning cells of individuals and producing cells or organ systems that are lacking in those individuals. And we can see that again. One of the things that we're doing is we're trying to get stem cells from the umbilical cord of individuals right as they're born. There's a number of stem cells inside of there, and we're trying to bank those and kind of use those, and that may be a possibility of them. Another possibility is that we see that cancer is basically a genetic disease. It's not really differential that grows. And so basically what's going on here is we have this proto-oncogenes, and these are genes that have a function in development of the cell. And what can happen is we can mutate these genes and that they become oncogenes. Again, these genes are responsible for normal growth stimulation. These genes are responsible for turning a gene off. And what we've done with a bunch of these is that we've done some sequencing. And so we've looked at many tumors located in various portions of the body, and what we see is that these tumors will have their own sets of genes that are turned on or turned off. And so a big problem with us being able to produce chemicals that cure cancer is that for a long time we were looking at cancer because of where it was at, not the genes that were turned on and off. Because nowadays we can do sequencing of our genes, of these structures here. We can look at those particular mutations. And so again, looking at colon cancer, we can see what it was. And this one here is looking at colon cancer. When we look at colon cancer, it's a series of mutations, 2 or 3 mutations that are required to produce a cancer cell, not necessarily one cancer. And so it's our exposure to cancer-causing agents, carcinogens, that cause cancer, and not necessarily other thing. And this is a list of all the different cancers that we have in here. And so that's basically what we have. And I will